Live from New Hampshire, it's time for the Science Cafe!
Learn about the latest technologic innovations today.
And you don't have to pay.
Expert panels with advanced degrees are sure to satisfy your curiosity.
Did we mention it's free?
And if you think you'll find a better time than this, then come on down and test your hypothesis. Cause you're gonna learn a lot if you wanna stay down at the Science Cafe.
Good evening, and welcome to our February 2021 episode of Nashua's Science Cafe of New Hampshire. My name is Rick Irving. I'll be your host and moderator for this very timely session on COVID-19 vaccines and vaccinations. Now, after nearly 9 years and over 100 science cafes, which were conducted in person, involving hundreds of panelists and thousands of attendees, starting last May due to the COVID-19 pandemic, we've gone virtual. So this is our 8th virtual session. And as we have done for all of our previous meetings in the last 5 to 6 years, this one will also be recorded and will be available for viewing immediately afterwards on Facebook and YouTube. Tonight, we're here to talk about the COVID-19 vaccines and vaccination process. Here in New Hampshire, we are currently in phase 1B, which means that all residents ages 65 and older can sign up for COVID vaccination. But shortly, vaccinations will be available to everyone, and tonight we want to explore the choices we will all face. Clearly, to end the pandemic requires sacrifice, but hesitancy to vaccinate is one of the threats to the plan to reach herd immunity. It's important to address these concerns and help people develop the comfort and trust required to get vaccinated and help us all get back to a more normal existence. Science Cafe New Hampshire is proud to bring you this chance to talk with real experts and get scientific answers to your questions. Science Cafe always focuses on bringing the public together with experts to foster an educational discussion based on science, facts, and reason. Civil dialogue is our goal, and the key to the interaction is the questions submitted by you, the audience. So feel free to use the comment section of Facebook or YouTube to send in your questions, concerns, or ideas at any time starting right now. Please be succinct, and we will try to get to as many of your questions as we can in our allotted time. So now I would like to take the opportunity to introduce you to tonight's panel and also give them a chance to tell us a little bit about themselves. First, we have Jennifer Swabowicz, who's an RN. She got her BS in nursing from St. Anselm's College right here in New Hampshire. And currently she is the immunization coordinator for the Nashua Public Health Department. She also has quite a history in the nursing field. She was a nursing case manager, helping clients awaiting organ and stem cell research transplants. And she was also a pediatric nurse for quite a while. Jennifer, welcome. Nice to have you here.
Hi, great. Thanks for having me. It's great to be here.
Great. Tell us a little bit about yourself.
So, like you said, I'm the immunization coordinator for the City of Nashua, the Department of Public Health and Community Services. I started in July, excuse me. I started in July, kind of right during the pandemic, and kind of jumped in with both feet. At the beginning of this rollout, it was still important to keep all our other immunization programs going with our outreach, and especially at the beginning of the fall with the flu vaccine. So lots of those clinics, but more recently we have begun doing some small COVID clinics in the community, reaching out to those that are unable to get to the fixed site, through VAM. So elderly housing in other communities that are in need. Prior to that, like you said, I was a case manager, but the bulk of my career, I was in a pediatric office here in Nashua. So I was the immunization coordinator in that office. So I have a long background dealing with vaccines and immunizations, mostly with childhood vaccinations, but, I've learned a lot through the years about all vaccines.
Great. Thank you so much for joining us tonight. And next we have David W. Clarke. David has a PhD in toxicology from Queen's University in Ontario, Canada. His expertise is in toxicology and pharmacology. He is retired from the Pfizer Corporation, where he was involved in developing vaccines. And one of the pluses of having to do these sessions virtually is we have a wider reach, not only for our audience, but also for our panelists. So David is our first international panelist, and he is joining us tonight from his home in Toronto, Canada. David, tell us a little bit about yourself.
Hi, thanks, Rick. Thanks for having me. It's a pleasure to be here. So I'm a really toxicologist by training. Spent about 30 years in the pharmaceutical industry. 10 or 12 of them, I guess 12 or 13 of them were spent in upstate New York, a place called Chazy or Plattsburgh, New York. So I'm used to sort of the North Country and life up there. For the past, and that's when I sort of started getting involved with vaccines. And for the last 10 years, when I was working at Pfizer, I was responsible for sort of the non-clinical safety evaluations of all the different vaccines within the Pfizer portfolio. So there were numerous vaccines for infectious diseases, therapeutic vaccines. They had one for smoking cessation, a number of different cancer vaccines that they were working on. So I've sort of seen a lot of the modalities and the technicalities and how sort of a vaccine is developed from the beginning through right through to registration. And so I retired, I guess, about 2 years ago. But I hopefully can answer some of the questions on perhaps some of the more technical aspects of vaccines and sort of what they've done. I don't, what I'm getting from the Pfizer and the products are really what's in the public domain, but hopefully I can sort of make them a little more understandable to people. And I thought to begin this that I put a few slides together to maybe talk a little bit about what vaccines are, and sort of what the different modalities of vaccine, sort of what we've seen up to date with the COVID vaccines. And just sort of to set the stage. And because with the COVID vaccines, I think we're seeing some really different vaccines from what's traditionally been out there. And it may be important to sort of understand some of these differences. And there are sort of three buckets that are in late stage for vaccines for COVID. And the first ones I'm going to call nucleic acids. And these are really the two that have been approved are RNA vaccines. And so the whole point of vaccination is you sort of precondition the immune system so it recognizes something on the virus or bacteria, mounts an immune response to this protein usually on the surface. So then when you get exposed to the live virus, your body is preconditioned and is able to quickly mount an immune response. I want to point out that, for RNA and DNA vaccines, we're giving, they really only contain a very small portion of either RNA or DNA, enough to encode for, in these cases, the spike protein, which is that protein that sticks out of the surface of the COVID virus. And so both of these, when they get injected, they get taken up into the cell. They borrow the mechanisms within the cell to make the viral proteins. Those viral proteins then get exposed or presented on the surface of the cells in your body, and that's what elicits the immune response. But because it's only that, and the RNA or the DNA quickly gets broken down afterwards, and eventually these the proteins on the surface will get broken down. They'll be there long enough to elicit that immune response. So this is the Pfizer, BioNTech, and Moderna use an RNA. There's a company called Inovio that uses a DNA vaccine. Same sort of idea. It's just one's giving DNA, which then goes to messenger RNA to make the protein. With the RNA vaccines, the RNA directly gets translated into the proteins. Another type that's out there is the Oxford, AstraZeneca, or the J&J, and these are viral vectors. And here they're using a non-replicating viral vector that contains DNA, and much like a nucleic acid vaccine, that viral vector is simply a delivery mechanism to get the DNA into the cell. The cell converts the DNA to RNA, RNA to protein. That protein gets expressed on the surface, and that's what creates the immune response. The last type main type that's really in development is a protein-based, which is Novavax, Sanofi, GSK were doing this. And in this case, they're actually making the protein, growing the proteins, and injecting that. The protein gets taken up into the cell and gets directly exposed or presented on the surface of the cell to start that cascade. And I've got a slide a little bit later on about the cascade of the immune response. So if we can go to the next slide. So just to give you a sense, here's the RNA, the technology RNA, and sort of this is I stole from the Wellcome Foundation. So it's suitable for people with weak immune system. The number of doses, so there are some differences. A lot of the ones we're seeing now are 2-dose. The J&J, Johnson & Johnson one is only a single dose. I think we've probably all heard there's, particularly with the RNA vaccines, RNA itself is rapidly broken down in the blood. So if it's injected as native RNA, it quickly gets broken down and would be ineffective. So they put it in a lipid, and that protects it, both protects it from degradation in the blood and also helps it get into the cell. The challenge with that is the storage conditions. So once it's in that lipid, it needs to be stored in cold conditions. And really, at this time, there are no other licensed vaccines that use RNA technology. Viral vector is Oxford AstraZeneca, Johnson & Johnson, has better storage conditions. There is one approved, and that's for Ebola vaccine. I haven't talked about the whole virus. This is more, none of these, in my opinion, at this stage are imminent for approval, but this is where you sort of inactivate the whole virus. Again, probably 2-dose, better storage condition, and there are lots of examples. So this is a more common technology for vaccines. And then the last one is that protein subunit 1, Novavax. Again, 2 doses, nice storage condition. It usually is stable, and it's similar to what we see with the hepatitis B vaccine. So if we want to go to the next slide. So here's a quick overview of this one. And it's a little complicated. I'll try to go. So you deliver the vaccine within these specified or specific cells in the body, antigen presenting cells. The vaccine gets taken up, the protein gets expressed on the surface, it gets recognized by T cells in our body. The T cells interact with B cells, and these are all immune cells within the circulation. The B cells then start to expand, they turn into a plasma B cell, and that's what creates the antibodies, and those antibodies are what's recognizing the virus. T cells also have, or these T helper cells also have the ability to go and form, transform into a naive killer T cells, which then can be activated, and those killer T cells can go and actually kill cells that contain that protein on the surface. And a key message with this is that both the B cells and the T cells, a subset of those will remain as memory B cells or memory T cells. And what's important about these memory cells is when you get reinfected, so this whole process where you go from antigen presenting cell to T helper to B cells to plasma cells, that takes time. It takes days for this to go and to be able to build up that immune response. Once you have the memory cells, it becomes a very rapid conversion of those cells into to produce antibodies. So this is why we see a very rapid response afterwards. And I think we have one more slide, hopefully. And this just to give you an idea, what about the safety? So Oxford AstraZeneca, this is one I took from the British Society, so it's US-based or Europe-based. They had about 23,000 people in their trial. Pfizer had 44,000 in their trial. Both of them gives a strong immune response. 2 doses for AstraZeneca, 4 to 12 weeks apart. 2 doses for Pfizer, 3 to 12 weeks apart. And here's just some of the common side effects that we saw with respect to these vaccines. Yes, you're looking at 70-80% of the people will have some sort of an adverse reaction. Typically, they were grade 1 or grade 2, so didn't really impact your ability to function. And I think typically when we looked at it, it was only lasting a day or two for the majority of these signs, and they're pretty typical of what you get with any vaccine. And one more slide, I think we have one efficacy one. Is it one? Yeah. So this is, when I saw this, I went, wow. So this is from the Pfizer presentation to the FDA. And so it was a randomized, 22,000 got the vaccine, 22,000 got the placebo. And this is the incidence of COVID occurrences in the placebo group in the red. And you can see how that goes up and up. And then the vaccine group is the blue line at the bottom. And you can see starting about day 14 after the first dose, those curves really start to separate. So at the day 14, day 12, not much difference between those. But by day 14, you're really starting to get an immune response and you're starting to see that separation. And this is really why they were able to achieve over 90% reduction in COVID with this vaccine. So I think that's the end of it, of my presentation. And I welcome any questions people have.
Great. Thank you, David. That was very informative. And I think we have some questions starting to roll in. This first one is here. What patient reactions have we seen in New Hampshire, and how is this data tracked and reported?
Well, I can certainly answer that.
Please.
The reactions we're seeing in New Hampshire are the same as we're seeing across the country with the vaccines. Typically, there's the pain at the injection site, which is very common and very common with all vaccines. But what, I've heard most is the headache, the fatigue, chills, body aches, and that after the first vaccine, you see mild reaction. After the second vaccine, which is 3 weeks out after Pfizer and 4 after Moderna, you're seeing those same side effects. They tend to be just a little bit more because your body has already seen this virus, is already ready for it, so it has a little bit more of a reaction to it, and it's that same chills, body aches, low-grade fever, headache, muscle aches. And I am hearing this lasts more than that, 24 hours. It could be 40, 72 hours. But nothing, personally, nothing that kept me from doing anything. And it was well worth it.
Great.
And I'm sorry, what was the second part of the question?
Oh, how is it tracked and reported?
So there is an app that if you get the vaccine, you can download to your phone. It's called VSafe. And it's a monitoring app. It texts you once a day for a week and then weekly for a few weeks just to check on your symptoms and see, what kind of reaction you had.
And is that app available like all across the country?
It is available all across the country. Yes, it is. For everyone that receives the vaccine, they should be given a form with this, and it has the QR code on it. You just, with your smartphone, you just, look up the QR code, and it's pretty simple to use.
Great. Okay, thank you. Now, what about internationally? Do we share that sort of data with Europe and Asia, or are they on completely separate tracking systems?
I'll take a stab. I think they're on somewhat different tracking systems. The companies themselves have a reporting requirement, and they should be tracking these in some nature as well. But I think that is one of the questions of how are we going to track all of the adverse events and the signs that we're seeing with these vaccines on a global basis. To me, it's unprecedented, the number of doses of vaccine that we're administering so quickly with this. I was looking up just the other day, the seasonal flu vaccine has about, I think, 170 million doses manufactured in the US a year. And, so that's for the flu vaccine. And we're looking for what, 300 million in the US for COVID?
Right.
So, it's a tough job to keep track of those. But I think there's a lot of different programs and techniques for doing that. And
Great.
Certainly, I think, and correct me, Jen, if you see a significant adverse response, there are reporting systems through the CDC for tracking those.
Yes. That's called the VAERS system. And that can be reported by the person that has the adverse reaction, or the doctor or the clinician that discovers it. But that's a system called VAERS, VAERS, and that tracks, adverse reactions, true adverse reactions versus side effects that are common. And that, right now, adverse, we see some anaphylaxis with the vaccine, like we do with any other vaccine or medication. But the research I could find today, Moderna, I think, had 10 true anaphylactic reactions.
Oh, wow.
In the first month out of, 4 million doses given.
Wow.
So it's fairly low, I would think, for the mass amount of vaccine that we're doling out at this point.
So when Pfizer was presenting to the FDA and the CDC, the UK had just presented or just announced that they had, I think, 6 cases of anaphylaxis associated with the Pfizer. So the Pfizer vaccine got approved in Europe or in the UK first, and they were immunizing people. And that, there's a whole series of questions through the FDA and the CDC about, what is this? Why are we seeing more? Is it just an anomaly that they saw in that first group of people? And I think early on we saw a few, but I'm not hearing much about anaphylaxis anymore.
I don't think the rates are that high. It's looking very safe.
Yeah. Great. Thank you. Next question. How would vaccines using different technologies, mRNA for Pfizer, Moderna, DNA for Johnson & Johnson, Sputnik 5, recombinant protein for Novavax work in combination?
So the big question is, in my mind, so they're all basically or hopefully eliciting an immune response to the spike protein, because the RNA vaccines are coding for the spike protein. The J&J and Sputnik are the spike protein, and the Novavax is the spike protein. So there should be some crosstalk. What we don't know, and we really need to do probably the studies, is if you get the first dose of Moderna, can you get the second dose of J&J or Novavax, and would that serve to boost? And I don't think we know the answer to that question at this time. I know there's been discussion about, particularly as supply is somewhat constrained, could you switch vaccines in the middle of the vaccination? And I think most of the regulatory agencies have come out quite strongly to say no, you should stay with the same vaccine, because there is no data that supports that switching in the middle of your dose regimen is still going to make it work.
Okay, great. Thank you. Scott, we have next question. From John Roxby, Elise Bernstein. What are the guidelines for behavior after receiving both doses of vaccine, as in visiting relatives, traveling, and so forth?
The guidelines for travel after the second dose of your all we have right now is the Moderna or the Pfizer vaccine in New Hampshire. But the guidelines are, if you are 2 weeks past your second dose, you do not have to quarantine after travel. But beyond that, we still need to do all our mitigation measures, such as, wearing our masks, social distancing, because we don't really know at this point if, say, you've fully immunized and you don't become sick, but you could still become an asymptomatic carrier. So if you're around people that are not vaccinated, you could still potentially, carry the virus and transmit it to somebody else. So at this point, the travel guidance has changed. Like I said, if you're 2 weeks post your second vaccine, you can travel, and when you come back, you don't have to quarantine. But beyond that, we really still need to be very careful with our mitigation measures.
So when you travel, do you have to follow some certain guidelines from state to state, even if you've had the second vaccine for 2 weeks?
I'm sorry, that cut out on me.
Oh, If so, when you're traveling, even if I've had your vaccine 2 weeks prior,
Yes.
do you have to follow guidelines in other states that you visit? So, for instance, Massachusetts may say you have to
Yes, you
isolate for
have to,
14 days.
whatever state you're traveling to,
Okay.
you need to follow their guidelines. I'm sorry, I should have clarified. This was New Hampshire guidelines. CDC has their own recommendations too, which are close to that. But you really, if you do travel outside the state or the country, you need to follow that state or country's guidelines.
Okay.
Okay,
Great.
we've been having these discussions because here the border between Canada and the U.S. is closed, and there's
Yes.
now a requirement that you have a negative test before you arrive at the border, and then another negative once you cross the border. And people are saying, "Well, if I've been vaccinated, why do I need the test?" And they're saying, "No, you still need the test."
They still there.
And
Yeah.
it's that reason of you may be an asymptomatic carrier and may still be shedding the virus to other people.
Right. Right. Okay. Great. Thank you. Next question from Stacy Samoji. Is it normal not to have side effects after the second vaccine?
I think they're meaning
Yeah.
after the second dose.
Yes.
Yeah, that's fine. I think that's normal. All bodies react differently. Your
Mhm.
immune systems all vary. So yes, it's I think okay, no matter what your reaction is, and if that reaction is no reaction, that is your body's immune system working.
Oh, that's probably ideal, I would say. Right.
Yeah, wouldn't that be nice?
Right.
Well, sometimes, but sort of the reaction that you get is showing that the vaccine's working, because that means, when you get this sore arm, when you get a fever, that generally means that the vaccine is starting that immune response and is doing what it should be doing.
Great. Excellent point, David. Thank you. And next question. I know we have several in the queue. I've seen them slip by. Could there be synergistic effect due to differing mechanisms, or could they cancel each other out?
Good question. I think probably unlikely that they would cancel each other out. Whether they'd be synergistic or additive, I, I would tend to think they might be more additive. So it's a what they call a prime boost when you get the two doses. And are you able to boost with a different vaccine or through a different mechanism? Essentially, the DNA, the adenovirus, and the proteins are all really acting the same way. That you're making the proteins in cells, or you're making that spike. You're getting the cells in the body to make the spike protein, express it on their surface to kick off the immune response. So the mechanism, even though the delivery is different, and how we get to that spike protein, the end result for all of those mechanisms is the spike protein being expressed on the surface of the antigen presenting cells.
Okay, great. Thank you. Next question. Where are the COVID-19 mutations occurring, spike protein, other proteins, that can cause a challenge toward the vaccine?
They're occurring all over the vaccine. We're only looking at this. We tend to be looking and making vaccines to the spike protein because it's the one that's on the surface, and it's the one that it uses to bind to cells and get taken up into cells. So they are occurring there. They are a concern for vaccines. So the one difference in my mind between a vaccine and a monoclonal antibody. So you've heard that people who have COVID have been treated with antibody cocktails. So a vaccine makes an immune response to multiple sites on that spike protein. It's not just one. It doesn't make it to the whole protein. It makes it to little pieces of the spike protein, but a bunch of little pieces of that spike protein. And so if one of those pieces of the spike protein mutates so it looks a little different, then hopefully there are other ones that will still recognize it. A monoclonal antibody represents one or recognizes one specific site on that protein, and always the same site. So a mutation in that site means your antibody will no longer recognize it. So there's a little difference. A monoclonal antibody recognizes one site. A vaccine makes a polyclonal response, so it recognizes multiple sites on that spike protein. So a mutation in one may not have as much of an effect on, its ability to recognize that spike protein. But as they pick up more and more mutations, then eventually you may get to a point where the vaccine no longer recognizes that spike protein, which is what the companies are looking at now with the different variants that are out there. Are they still going to be efficacious? And we have to remember that the clinical trials for the vaccines for the Moderna and the Pfizer vaccine, those clinical studies were all done before the variants were really largely in the population. And so, there isn't a lot of, we're getting some clinical data now on it, but, the data that went into the original approvals in December was really without the variant. And that's been the question is, will the vaccine still be efficacious against that variant?
Right. Okay, thank you. Next question is from Susan Belanger. My mom and aunt, both are in their 80s, had COVID and were both very sick for over a week. They only want the first shot, not the second. Would that be okay?
That wouldn't be the recommendation. The recommendation would be for them to both receive both vaccines, even though they've had COVID. I'm not, that second one really just boosts up. I know their system may already be a little primed for it because they've had COVID, but it really is important to get, both of the vaccines. The first, and the second one 3 to 4 weeks later, depending on which vaccine. It will really make sure that the system is prepared. Your immune system is prepared if it sees it again.
In hindsight is 2020. So they designed the clinical trials as best they could with what they knew. There's a whole bunch of other questions that they could answer. And these are, if someone had COVID, could they get away with 1 dose versus two? We don't know. It's all theoretical. Those studies have not been done to verify it. And so the recommendation, I agree with Jen, the recommendation for the health authorities is you follow the recommended dosage regimen for it. So if it's a 2-dose, you take the 2 doses. Mm-hmm. It's important to point out, you're not giving them COVID. You're giving them the spike protein. You're giving one little piece of the virus.
Right. So maybe reading into the question a little bit further, because her mother and her aunt had COVID and had the first vaccine and got sick, could they expect to get sick again, or that we just don't know what would happen if they get the second shot?
Everyone's different. So you never know. People who've not had it and had, get a reaction after the first, but not after the second. Someone get it after the second, but not after the first. People are very different, particularly with respect to the immune system and how they're going to react to a vaccine.
Great, thank you. Next, from John Glatch, we cannot create a vaccine channel through physician offices and local clinics like Dartmouth, for example. This would be convenient for many people. Is there a question? Oh, why can we not create the vaccination channel? Excuse me.
I'm not sure I quite understand the question.
I think to me, the question becomes, and part of the issue, so the 2 vaccines that are approved, Moderna and Pfizer, have storage requirements for the vaccine that most typical physicians' offices will not have. And so, to account for that, they've had to go to specialized facilities that have the ability. The other one is, I think they're both multi-dose vials.
Yes.
And so there's 5 or 10 doses in one vial. There's no preservative in that vial. So when you open them, to take out 1 dose of a 5-dose vial, you'd have to throw the rest of it away unless there's someone there to receive the next dose.
Right. to really move through the mass amount of people we want to move through it. It just doesn't make sense to do it in a small doctor's office at this point. We really need to do it at these bigger events, these fixed sites across the state, to make it more accessible to more people. It just doesn't work. And it also, bringing people that may be sick into a doctor's office while they're also bringing in people to get vaccinated for that particular virus, it really sets up for a logistical mess at this point when we're trying to vaccinate so many people. So I find it, the fixed site in the VAM system have been a challenge in New Hampshire, but they're really rolling out the number of vaccines. It's an incredible amount of vaccines we've been rolling out in such a short amount of time. We all want it, and we all want to get it as soon as we can, and the state is working hard on it.
Great. Okay, thank you. From Facebook, Michelle Beck, can vaccinated people still spread the live virus while not becoming sick themselves?
Possibly. We don't know. We haven't answered that because, we don't have the data to say yes or no. And that's driving the recommendations for, if you've had COVID, to get both vaccinations, because we just don't have the data to say one way or another whether that's true or not.
But the potential is there.
Yes.
Okay. Thanks. So I'm going to insert a question here. I think I've been given permission from my daughter to ask this. What about pregnant women and the vaccine? Should they be getting the vaccine? Does it have any effect on the embryo?
It's being studied. Pregnant women, breastfeeding women weren't in the original studies. Some were inadvertently in the study. And so far, we have seen that it is safe, but the recommendation is yes, they should get vaccinated. But I think any woman that's pregnant and has a concern should talk to their doctor and go over the risks and benefits of both to feel comfortable with it. But I do feel that the studies will show that it is safe.
It's the vaccination in pregnancy has been an issue for all vaccines for years and years, and it's really, now influenza. Even if you're pregnant, they're recommending that you get vaccinated with influenza for the seasonal flu vaccine, and I think we're getting more. There's getting to be more comfort with vaccination during pregnancy. But I agree, it's a discussion between the pregnant woman and her physicians as to the risk benefit of vaccination while pregnant.
Great. Okay.
It's not contraindicated at this point. So it's not saying you should not get vaccinated. They're saying have that discussion.
Okay, great. Thanks. From Tom and Lynn Bloomquist, how many double vaccinated people in New Hampshire have turned positive in Massachusetts? Not by regulation, by science.
I'm not sure the question.
Well, I think the rate of the COVID-19 in Massachusetts is higher than New Hampshire. So, originally there was some people from Massachusetts were not encouraged to go across the border, for example. So if somebody has been vaccinated, got both their vaccinations in New Hampshire, what are their chances of going and getting the virus there?
Well, to me, it comes back to that same question. If you've been vaccinated, can you still be a carrier for the virus? And so the end point of the clinical trial was COVID disease. And so, I think in pretty much all of the vaccine trials to date, we haven't seen, or we've seen very few, if any, serious COVID disease in people that have been vaccinated. But that's still not saying that someone who has been fully vaccinated will not get COVID or be able to shed COVID or may test positive in a nasopharyngeal swab for COVID. And we just don't know whether that means that they're able to spread the virus to other people.
Now, a lot of these unknowns, are they because we had to rush the COVID-19 vaccine into production and get it into people, or do all vaccines come into this same scenarios?
I think it's a question with all vaccines. There's probably a little less data because normally in a vaccine you would have run, more trials. You may
Right.
not have had more people vaccinated in those clinical trials because, the size of these trials at 44,000 or 23,000 or 60,000, I think for J&J, those are huge trials, and that's as many people as you would, total number of people exposed and most vaccines being developed. But it's all of these questions about these various scenarios that they weren't asked at the time, or that, because that would have slowed down the development process. And I think we're now trying to fill in those holes in the knowledge.
Right. Okay. Now, one of our staff here, Robert Samoji, the CDC says science supports reopening schools, but when Israel did that back in 2020, it led to an outbreak with both students and teachers infected. What does the CDC see differently?
I think they're seeing the importance of getting kids back into schools and the socialization and the learning that may have been lost with the pandemic. And they really have provided the schools with great guidance on how to prevent it, the social distancing. Here in Nashua, they've just gone back and it's a hybrid where, so maybe I think, in the high school, there might be a quarter of the amount of kids in the class at any one time. So I think the CDC is weighing the risk and benefit here of keeping kids out of school and what toll that's taken on them. But I do think on the other end, they have provided with great guidance on how to keep the kids safe and the teachers safe in the classroom.
That's why I want to get as many people vaccinated as quickly as we can.
Sure.
Right. Great
Sure.
segue.
So that you develop this herd immunity that they talk about.
Yeah.
And there's less of a risk. And I think some of the things I've seen coming out of Israel now, it's open. I think they're at 70% of the population is being vaccinated now, and they're starting to open things up. I think it's also important to see that the one report I said that they're seeing about the same level of efficacy for the Pfizer vaccine as they saw in the clinical trial, which I think is another important thing. Right. So in a real life use of that vaccine, we're seeing the same sort of numbers that they saw in the clinical trial, which I think is encouraging for the vaccines.
Yeah, I agree. That sounds great. Okay, well, we wait for the next question. This is a general one that I think people would be interested. Oh, here we have one from Robin R. Robinson. If vaccinated people can still pass the virus along, how can we achieve herd immunity? Also, ability to spread virus while vaccinated, normal or exceptional?
So it's another good question, right? If you're spreading the virus, why? So I think no. Usually, one hopes that eventually you'll get to a point, and that even these vaccines will be able to neutralize the virus, and you'll lower the viral loads that the person carries, so that yes, they may still be shedding virus at a very low level, and probably not enough to really reinfect more people. But yeah, no, it's a good question. We all talk about herd immunity, but if we're not getting rid of the virus completely, then how do we achieve herd immunity? And I think it's so there's early on, if you go back to last year, a long time ago, there was all this talk about, close proximity for 15 minutes. And, when they talk to the 6 feet, it's the total number of viruses that you see. And, why does this matter? And they had all of these aerosol experts talking about, how far it can travel. And And I think that's based on a particular viral load. And there's sort of a thought that if you can lower the amount of virus that you're exposed to, you'll potentially decrease the either developing COVID or a more severe case of COVID. But that's it. At this point, it's still somewhat theoretical, because we haven't done a study with people that have taken normal people, and what you do is you dose them with different levels of virus to see what they get, but no one's going to sign up for that trial.
Right.
Although,
Okay.
the UK has just started a challenge trial. So they've started a trial in the UK where in a controlled environment, they are going to give people, expose people to the COVID virus.
Really?
To see how the vaccine works. And those are the sort of questions that you can start to answer. Is there a difference in the amount of virus that's present? But remember, it took 30,000 or 20,000 people in these trials to get the data that we wanted to approve the vaccine, or so they're going to be huge trials, and they're going to take a while to run.
Mm-hmm. Okay, thank you. Go ahead. No, Jen,
I was just going to say, in these trials, the efficacy is 94-95% with these vaccines. So, I think there's really great hope that we're going to get to herd immunity, that, even if there is asymptomatic carriers, that the chance that the spread is very low for those that are maybe asymptomatic versus someone that's very sick with COVID going out in public and spreading it. I think it's the asymptomatic carriers. If after you're vaccinated, you can become an asymptomatic carrier, that's still an if and being studied. I think the chances are low, and we can get to herd immunity, but that's going to take 70 to 85% of everyone getting vaccinated. And because we're such a transient world, it's going to take the world getting that amount of vaccination out there, not just the US.
Right, that's a great point. Okay, next question from Sandy. When thinking about allergies and this vaccine, what type of allergies may be of specific concern?
So with both the Moderna and Pfizer, the biggest one that comes to mind is something called polyethyl glycol and polysorbate, which I think is more the
lipid nanoparticle.
Yeah, that is encasing the vaccine. So those are definite contraindications to getting the vaccine. If you've had an anaphylactic reaction to a previous Moderna or Pfizer vaccine, that's a definite contraindication. But otherwise, all other
I lost her.
Jen, we just lost your audio for a second there.
Sorry, can you hear me now?
We can, yes.
I'm sorry. So, but all other allergies at this point, even if they're anaphylactic allergies, are precautions. And, at all the sites, they'll keep you 30 minutes just to keep an eye on you.
Right.
15 minutes for people without allergies, but 30 minutes for those that have allergies, just to keep an eye on you and make sure, that there isn't an anaphylactic reaction.
And the fortunate thing with anaphylactic reactions is if you're being watched, they can do something about it.
Yes, we have epinephrine right there.
Right. So they're ready for it.
Yes.
So even if it does happen,
it's very treatable.
Now, the recommendation if you have anaphylaxis with the first dose is you would not go for the second dose.
Right.
Okay. Yeah. That makes sense.
And there's one with these that they're seeing a delayed. My mother had got her Moderna vaccine in the end of December and got a rash about 7 days after her first vaccination. And she got a rash on her arm.
Right at the injection site.
The injection site lasted 2 or 3 or 4 days.
Yeah.
Didn't hurt. And
not a contraindication.
No, she got her second dose and it was fine.
Yeah.
But they are seeing these delayed rashes coming on, 5, 7, 8 days after you get that dose.
I've heard that a few times, actually.
Yeah. Okay. Next question from David Mills. What is the harm or downside of having Pfizer for one dose and Moderna for dose 2 or vice versa? We don't know, I think.
Yeah, we don't know.
Right.
We don't know the answer to that. And I think there was I thought actually the UK, someone I thought was going to try to run a trial to look at that, but they haven't done it yet. So the one thing that we're not sure about is they talk for some vaccines, they have what they call a correlative protection. So that if you can see if you get a certain antibody response with the vaccine, we know that's protective. And we haven't established enough of this yet to say this is the correlative protection that you see. Flu is one where you can say, okay, here's your correlative protection. If you reach this level, it's most likely going to be efficacious. Once you get things like this, then you can talk about flipping back and forth and doing some other things. But we're not there yet. We haven't had enough experience with this and haven't generated enough data. It's been, when was the first case reported in New Hampshire? 12, 13 months ago? We're pretty quick into this, that we're talking about this.
Right. Okay, thank you. Next question. The Pfizer and Moderna vaccines underwent clinical trials with specific times between first and second doses. Yet some people say that time can be extended in order to get more people the first dose. What is the science behind that, and has that been proven for mRNA vaccines as well?
So great question. Pfizer is 3 weeks between dose. Moderna is 4 weeks between dose. When the Moderna was presenting at the FDA, they were asked that question. Well, why are you 4 weeks and Pfizer's 3 weeks? And they said, well, it's what we ran the trial. I said, that's sort of in the vaccine world, you give a prime dose and you give a boost dose 3 or 4 weeks later, and they picked 4 weeks. And again, that's what they picked going into the trial. That's what their data is from. And so that's why they're recommending 4 weeks between doses. Now, when you run a clinical trial, everyone doesn't get it at day 1 and day 21 or day 1 and day 28. Some people can't go in that day, so they come in early. Some go in late. And so there's a range of time between when that second dose is given relative to the first dose. And I think they've gone back and looked at some of these, and the health authorities are recommending that you follow the regimen of 3 weeks or 4 weeks. There can be some leeway in those time frame. And I think as vaccines have become shorter or in short supply, some regions are saying, no, we can go out to 4 weeks or 5 weeks or 6 weeks or 7 weeks. So there's no hard and fast rule as to why it's 3 weeks or 4 weeks. It's somewhat empirical. This is what's worked, and that's how the trials were designed, and so that's the data that they have to support that 4 weeks. They don't have data. When Moderna doesn't have data that says if you go at 5 weeks, you won't get 95% protection.
Okay, so Jen, when somebody comes in and you schedule the first vaccine, you at that time say, okay, we want you to come back in 3 weeks. Do you give it
Yes.
and those sorts of things? Oh, that's great.
And what happens if they don't come back right on that day and they come back 3 days later?
We're still going to give it to them.
Yeah.
And, I think at any point you're going to do that second dose. If it's beyond, I think, there's a 4-day grace period to get it early for either the Pfizer or the Moderna. After, we're going to do it we want to do it at that 3 week mark or 4 week mark. But if we don't, and they come back 2 weeks later, 4 weeks later, we're going to give the vaccine. And, time will tell about efficacy, but in the past with other vaccines, it's, we don't start the series over, and it's proven that other vaccines have had good efficacy. So in time, we will know more. But no, we want it at that 3 and 4 week mark, but we will give it whenever we get them.
Okay, great. Thanks. Our next question is from Gail Taylor. After I have my second dose, can I visit another person who has also had their second dose?
I think I heard Dr. Fauci talking about this. You know, yeah, I think people still need to be careful because we're still learning so much about all these vaccines. But yeah, I do think, I think there's going to be some safety reports coming out on people that are fully vaccinated and both are 2 weeks beyond their second dose. I think we still need to be careful, but I do think you are in a safer circumstance than if you were with someone that was not vaccinated.
Okay, so you would expect that you would like people to still like wear their masks, for example,
They
things
should,
like that?
yes.
Okay.
Wear your mask, social distance, do all of it until we know more, but that
Okay.
is the hope.
That is the hope. I know a lot of people that have gotten their first one and they're dying to get the second one.
I know everybody wants a hug. Everyone wants to be with their family. I know, but I think
Right.
we still have a little bit longer to go on that.
Okay. Okay, the next question, Ray Burko. Oh, hello, Ray. Haven't seen him in a while. Do you anticipate having to have an annual shot of vaccine? And sort of aside beyond that, why are some vaccines annual and some lifetime?
So it comes down to those memory cells and how long they stick around for. So those memory B and T cells that when you get that vaccination, and you activate those B and T cells, some of them stick around as memory cells for long periods of time. And what we don't know is how long for the COVID vaccines they're going to stick around for. And so we don't. So it's how long will that immune response last? And that's the big question at this point. So if you look at the phase 3 trials, the phase 3 trials were designed to follow people for 2 years after their last dose.
Okay.
They got the emergency use authorization based on 2 months data from 50% of the subjects. So those phase 3 clinical trials are still ongoing. And that's where you would generate this data that says, oh, the immune response lasts a year, 2 years, 3 years. We don't have that data at this time. That's the challenge. And so we can't answer that question, or we don't have the data now to be able to answer the question, are we going to need to get a COVID vaccine on a yearly basis?
Okay.
Remember, COVID just came up a year ago.
Right.
And if it's somewhat real time that you're generating this data, and so it's going to take a year or two until we have a really good answer to that question.
Right. Yeah, it was almost exactly a year ago. I think March 13th, New Hampshire sort of shut down last year.
Yeah.
Just before my golf season started. Next, how do viruses mutate, and what are we doing to identify strains in New Hampshire?
Do you want me to do the why they or how do they mutate?
Sure.
So it's like any cell. So when the virus gets taken up into the cells in the body or in the lungs, et cetera, it multiplies and replicates within those cells. Every time it replicates, there's a potential for a mistake being done in that process, and that forms a mutation. Some of those mutations might kill that, the son of the virus that's generated, but other ones, the virus can still continue, can still replicate, except now the protein's a little different from what it was the previous time. And that it's just because it's replicating so quickly in so many copies, these mutations just build up in the virus.
Okay. And Jen, do we track these mutations in New Hampshire?
Yeah. The labs, some of the labs are doing some sequencing, genetic sequencing on the virus, and the nasal swabs they get at the state level. I'm not sure the rate of how many they how much sequencing they're doing. I think if it's a more suspect test, say someone's traveled outside and they know that in the last 2 weeks, or there's more of a cluster seen, I think they're more apt to do the sequence testing on each sample, but I don't have exact numbers. But I do know we're doing more sequencing now than we have with all these new.
Okay. Next, from Carol Isk. If someone doesn't have any reaction to their second dose, doesn't necessarily mean they're not getting an effective immune response.
No, they're lucky. But
That's the preferred response, right?
yeah, but no, they're still, their immune system is still recognizing it and seeing it, and maybe it's because they've had COVID or seen it before, and their body was ready for it and responded appropriately.
If you look at the controlled clinical trials that they ran, there are people in those trials that didn't get any response to the vaccine, to the vaccination. So there are people that are fortunate that don't get the reaction.
Great. Yeah, I'd say that would be the preferred response for everybody, I would think.
Yeah.
Okay, and this may be our last question. There are ongoing clinical trials regarding people who are not currently able to be vaccinated, pregnant women, children, and will it become standard for newborns in the future? Good question.
There are ongoing trials looking at pregnant women, looking at coming down in age with children. Whether it will become standard in newborns, I don't know.
I mean, flu vaccine is only as low as 6 months old. So, there are other vaccines that newborns do get at birth, but at 2 months. But flu vaccine itself is only as young as 6 months. So, time will tell.
And typically for these clinical trials is they go down in age. So you start with adults, then you go sort of to 16-year-old, then you go to 12-year-olds, then you go, a little younger to single digit 6-year-olds, 8-year-olds, and before you get to infants. So they do tend to take to make sure that it's safe and efficacious in sort of the older age groups as they go down in age. But I'm sure they're looking at it. Or will be looking at it. I don't think there are any trials started in the very young as of this time.
I think I heard yesterday in England they just teenagers and in that range of folks. But yeah, I've never heard of infants at all.
There are trials going for teenagers, even in the US, I think.
Yeah. So this was one of the controversies between Pfizer and Moderna. So Pfizer its emergency use authorization is for age 16 and up, and Moderna is 18 and up. And it's because Pfizer included people down to 16, a small group of them, in their clinical trial. Right. And there's a whole discussion at the FDA's advisory committee. So this is the committee that sort of experts that advise the FDA on the the quality of the data and the safety and the efficacy. And a lot of them were sort of saying, I'm not sure 16-year-olds are the right ones and we can really approve it for 16. And so if you look at they vote at the end, and there were 3 or 4 people that voted against the Pfizer vaccine, primarily because of the 16-year-old requirement or age limit on it. I don't think there's any difference in a 16-year-old and an 18-year-old, but it comes down, you have to have the data to support your indication and what age groups you want to go in.
Okay. Well, we've gone over our allotted time by just a few minutes, so we'll close out the program by saying, first, I want to thank the audience for tuning in and submitting some great questions. And then secondly, please join me in extending our sincere thanks to both Jen and David for generously giving us both their time and their experience. You have made this a great show, and I really appreciate it.
Thank you for having me.
Thank you for having me. Yeah.
Good. I hope you had fun.
Yeah.
Next month, our topic will be online privacy and digital security. So please join us on March 17th, which is St. Patrick's Day, for that session. And then lastly, before we sign off, I think it's appropriate for this topic that we at the Science Cafe of New Hampshire would like to express our sincere thanks and gratitude to the first responders, the doctors, the nurses, the healthcare staff, police, fire, EMTs, National Guard, people that do their groceries and transport the foods. I mean, given how difficult it was for 2020, I can't even imagine how much worse it would have been had it not been for those people stepping up and doing their jobs. So thank you to all of them. Really, really appreciate it.
Absolutely.
Absolutely.
Yeah.
Yep.
Thank you guys for joining us, and thank the audience as well. That is it for our program this evening. So everybody have a great evening and a good night, and we'll see you next month. Bye.